It's good to be here. I'm Tom Mumford. I'm not going to do a long introduction, but just to say, I was hired by the Department of Natural Resources in 1976 to start a seaweed aquaculture industry in the state of Washington. And as they say, the rest is history. So I've got a little track record here, but I'm here today just to moderate these two folks here. So let me go, if this is going to work. They're working for you back there, Terry? I just was commenting, how many of these kind of shows have I been in that just devolve into a mess, technically, and so far we're doing just great. Anyway.
Just jinxed us, thanks.
I just jinxed you what?
Jinxed us.
Just jinxed us? What happened? That hasn't happened yet. Anyway, so yeah, again, this discussion is about genetics, and I added diseases because they're kind of intertwined in many ways here. And we've got two folks to come help about this. But I just wanted to say that these are two intertwined topics. They're not separate at all. And there's a great interest in the genetics, not only in seaweed aquaculture, but just aquaculture in general. And I will expand that to say agriculture in general, that the development of and interaction of these organisms and the genetics of these things are of huge importance. The relationships between the wild stock, and we've already alluded to this, and population stocks, introduced species, you got gene flow between all these different types of organisms, really important kind of stuff here. From a practical standpoint, you get into sort of species selection, strain selection, stock source, availability, all these kinds of issues are things that we'll talk about here today. The diseases, likewise, I always had the quote, nature abhors a monoculture. As soon as you start growing a lot of one thing, there's going to be something that's going to take advantage of it. How do you prevent that? How do you control that will be a topic of some of the discussion today here. So the format, basically what I want to do here is talk a little bit about these two topics here. Keep in mind the kind of outline that Meg outlined this morning. We're here to coordinate, cooperate, learn how to move forward. This is in a positive way with all this kind of discussion here today. So each speaker is going to just say a few things about themselves here, and then I've got a list of sort of pre-arranged questions that we can go through as we see fit, and then at the end we'll open it up to questions from the, or not at the end, but at some point we'll open it up to discussion and questions from the audience, the participants here. And then I'll try to make some kind of a summary statement if I have my wits together at the end of all this. Okay? So, I'm the moderator, and Kate and Simona here. So, Simona, you want to get up and do this, or shall I? It looks like you've got a...
I can.
You've got a... This is a attitude thing.
Yes. All right, well, my name is Simona Augyte, and I just want to say thank you so much for Meg, Terry, Nicole for putting this together. It's really good to see everybody come together, various stakeholders, and talk about this. So I just wanted to share a little bit of my own phycological journey. And it all started, so I was at Cal Poly Humboldt doing my undergraduate work. And then I went on to do a master's with Frank Shaughnessy, where I looked at basically the marine flora between Cape Mendocino and Cape Blanco. And so we were interested in just figuring out what diversity was out there in between the two capes, and if there was differences at the capes where there was a lot of wave exposure. And we also wanted to kind of compile this with historical records to just have a baseline of information. And it was at this time that I went to a seaweed conference, and I actually talked to Bob Walland from UW, and I said, hey, I want to do seaweed aquaculture. What do you recommend? And he said, there's this awesome person called Charlie Yarish, and he's on the East Coast. And so I reached out to him, and I ended up working with him for my PhD. And coming from the West Coast to the East Coast, I found that there was low kelp diversity specifically. So, you know, there's a lot of sugar kelp, but basically where we were in Long Island Sound, it's the it gets really warm in the summertime, so it's kind of like the southernmost boundary for sugar kelp. So I went up north more and worked with this. It was considered a forma of sugar kelp, but based on some genetic evidence, we showed that this is actually a different species. So this is Saccharina angustissima, and we worked to kind of elevate it to that species level using genetics, but also we were the first to domesticate this species. And so now there's a lot of seaweed farmers in the area that are growing this cultivar, and they found that it's high yielding and it responds well to any kind of disease outbreak. So it's very resilient, which has been really exciting. And again, this was done at University of Connecticut. I worked in industry after I graduated for a short period of time, and then I went on to do a postdoc at UConn, and that was with Woods Hole Oceanographic Institute, and that was funded by Department of Energy Mariner Program to do seaweed farming offshore, but the emphasis there was doing kelp breeding, so developing cultivars and moving away from collecting wild stock, but developing cultivars in the lab and, you know, holding on to those gametophytes, making crosses, and then looking at the lines. So looking at genotyping and phenotyping. So looking at, you know, if you have a parent that looks like this and a parent that looks like this, you put them together, what's the offspring going to look like? And is that going to, you know, how does that, in subsequent generations, do you have the same traits expressed? So those folks are still doing that work, and it's super important. Another thing that we also did while I was out there is looking at the population genetics of sugar kelp in the region. So again, it has the southern limit around Long Island, but then we saw that there was actually a genetic break at Cape Cod. So the populations in the south were, you know, genetically different than what was found in the north. So to be conservative, we recommended that you wouldn't, you know, you wouldn't cross those two populations. So you would stick with the populations in the south, and then you'd stick with the populations in the north. But then I figured I wanted a little bit more sunshine, so I moved out to Kona. So I'm currently residing in Hawaii on the Big Island, and I work at Nelha for a company called Ocean Era. So I'm the lead scientist there, focused on macroalgae cultivation. And we are primarily also funded by Department of Energy, who are interested in cultivating seaweed for biofuels, food, and feed. And for now, we're doing all the work on land, but the hope is to go offshore. And the biggest challenge in working in the tropics is that the water is oligotrophic, so it's very low nutrients. And but the upside is that we have plenty of sunshine and we can cultivate year-round. So there's those things. And we are focused on cultivating different species. We have a couple reds, a couple greens, and no kelp whatsoever. So there's that. And on top of that, as a side project, I just want to put in a plug, I also co-founded the California Seaweed Festival with a colleague of mine that was also initially started with Sea Grant funds. But we also basically wanted to build a social license in California because there is a lot of interest both in seaweed farming, but also in just, you know, celebrating the diversity and celebrating the ecosystem services that seaweeds provide. And so we're putting, this is our third year. We had the festival where we wanted to bring people together from different disciplines, so both scientists, entrepreneurs, and artists, to kind of start talking and to see where there's an intersection in the seaweed world. So there's my plug. Thank you.
Katie? Hi, everyone. I'm Katie Davis. I use she, her pronouns. And I am the lead biologist with the Washington State Department of Fish and Wildlife in the new Shellfish and Seaweed Health and Biosecurity Unit. And so this is a newly funded and newly established or broadening program within the WDFW. And just to give you some of my background, so I came to DFW from a postdoc that was funded by the Hackeye Institute and the University of British Columbia, working on sea star wasting disease out at the USGS lab on Marrowstone. And so thinking about sort of marine diseases and what causes them and how they spread, and another important keystone predator that can impact kelp. And then for my PhD, which I did at the University of British Columbia, I studied the microbial communities that are on cultivated kelp, so kind of working towards some of this baseline knowledge to understand what healthy microbes are on cultivated kelp. And I also did some work on other intertidal algae and shellfish. And so that's sampling the microbiome of cultivated kelp. And then before that, I got to work on some projects with the Port Gamble Sklallam tribe, and I was a farmer and research technician for the Organic Seed Alliance on the Olympic Peninsula, and sort of working in the stewardship of organic seed and farming practices and on-farm selection and breeding, so sort of the terrestrial agriculture side of some of the topics that we're discussing now for kelp, and sort of thinking about local and regionally adapted varieties and varieties best suited for sustainable practices. So yeah, that's some of my background, and I'm really excited to be chatting with these experts and participate in the discussion with all of you.
Great. A lot of depth here, folks. Let's take advantage of this. So what we had done was to come up with a list of sort of starting point questions for you two. And you're going to have a hard time seeing this, I suspect. You're good? You're good? We know you're good, but anyway, so these are some of the questions that we kind of came up with. And I asked these two just to kind of answer, ad lib their way through these things. Some of these are going to be relevant and others in the give and take. So such things as for both genetics and diseases, what are your main concerns? What is the scientific basis for these concerns? In other words, is there something you can actually talk about? What science may be needed to further focus these concerns? And I'm saying, I'm taking, for example, like, we're worried about genetics. Well, what about genetics? And what part of that is of concern? And then how can we answer, or what do we know, and what can we do to get a better answer for that thing? A lot about what's known elsewhere. I think we've all been down this path. There's a lot, particularly in these areas, that have been done in places like Korea and in China and in Europe to some degree. And then how do the Washington regulations either address or not address these? And Katie, you're going to answer some of this. I think later on, Chris Adler is going to be here, or is here, and maybe could help answer some of these as well. What kind of short-term solutions might we have? What kind of BMPs might we start off with? And then, as we talked about, I think Emily and Meg both have put out very well, how do we move forward? What's the sort of cycle of adaptive management and moving forward and doing research and getting the answers and coming up with better BMPs, better practices? So with that, we've got a fair amount of time here, so let's just start down the list. Anybody want to start off number one here? And we'll start with genetics. Let's start with genetics. Got a microphone there, or you can come up here if you want. If you want to come up here and talk, you can see the mouse.
I'm okay to just sit and talk. Well, the first thing I just want to say is I think diversification is super important. I think I've seen a lot of interest here in just focusing on kelp. And kelp is fantastic, but I do think that there's an opportunity to think about other species as well, and maybe even, having a summer crop to diversify, or, intermix into your winter crop just so you have the habitat there. You know, if you have your lines, it depends on the permitting too. If you can keep your lines in the water all summer long, you might think about putting in another species that, you know, could also grow there. And then thinking about genetic diversity also, so definitely want to consider species that are native to your region and developing cultivars that are specific to your location. And I think this can become a problem, though, with climate change and range retreats, right? So that's something that will need to be addressed. But, you know, working within your region, I think, is obviously super important. You don't want to import stuff from maybe Southern California or something like that if you don't need to. So start with that.
Yeah, and that's a good segue, and I'll put on my DFW hat and just share the mission of the agency, which is to preserve, protect, and perpetuate fish, wildlife, and ecosystems while providing sustainable fish and wildlife recreational and commercial opportunities. And so our regulatory responsibilities are more in the prevention and management of pests and diseases and non-native species. But of course, with that comes preserving biodiversity and genetic resources, because loss of that can really increase susceptibility to disease. And so again, thinking about what biodiversity exists, what seaweeds are out there, and then the genetic variation within species and the population connectivity, so how gene flow is happening in the wild. And that can be useful in informing sort of management, like Simona said, thinking about how seed stock gets moved, transferred within the state, or where it's being sourced from, to be using sustainable best practices. Yeah, and then again, thinking about also best practices for restoration and producing seed stock, or things like biobanks, like PSRF is working on, and how we best manage those to really maintain genetic diversity.
Tom, would you like to add to that?
You know, this, you're,
You don't need a microphone.
so yeah, I'll just ask a question. So the question is, like on the genetic biodiversity kind of things, what concrete things could we be doing in the short term and long term, and what do you think the concerns are about if we don't do these things? We're still kind of thinking at a fairly high level here.
So if we're not working towards preserving genetic diversity, is that, what are the concerns?
Yeah.
So I guess we can draw from examples. So we're in a really unique position to draw from examples from other countries and other states, and thinking about places where maybe seaweed cultivation has moved forward without some of these considerations that we're discussing. And so in places like Chile, where like with agarophytes, there's been a strong reduction in genetic diversity because of the propagation methods or how they're sourcing their genetic material. And so that leads to all sorts of issues with disease and pests. And so I think that's a major concern.
Yeah, and obviously, you know, not native species introduction is a huge issue. So, you know, working with native populations is critical. And obviously, you can leverage the population genetics work that has been done for bull kelp in the area. So, and potentially work with additional genetic work. So if there is a lot of interest in, you know, maybe doing sugar kelp here, then working with scientists to get the population genetics analyzed and then seeing, is it okay to sample from different locations, or would you pick just one location because otherwise it's all the same? So I think that's important to think about. But there's a lot to do also in terms of like cultivars. And if you are to develop a seed stock, there's, people have bred sugar kelp to be disease resistant to be disease resistant and to be high yielding. So if that is a goal, if we want to grow disease resistant kelp, that is something to think about as well, and how to do that sustainably and have a seed bank that from that region, and then you cultivate your seaweed. So I think there's a lot of potential there.
I was curious this morning, the comment was made somewhere about Maine, and they haven't developed warm temperature resistant cultivars. And I think this definitely blends over into the restoration business, because I think we're going to end up having issues with global warming here. We already have, I think. And so, but Maine said you can't use those. And so how does that play into what we're doing here? Is this something we would want to do the same kind of a thing, or do you think we should move forward and be thinking about using those kind of cultivars?
Yeah, and that's an ethical issue. I know in Australia they've had huge kelp die-offs, and they are doing some restoration work, bringing them from other regions just so that there is that habitat, and that habitat, is there for the organisms and everybody else that needs those ecosystem services. So that's something to think about, is, bringing species from the south. But that's only if you need it, right? So if the species are tolerant, then just work with what you have for now.
Yeah, and I'll just add that I think in moving forward with something like that, one of the needs that we have is just a better understanding of what the existing diversity is and across species, and also one of the big questions is how does genetic diversity move between cultivated seaweeds and wild populations? And so we want to ideally prevent that from happening, because that's a way to lose genetic diversity. And so I think just thinking about developing more of those baselines will be really important moving forward.
So another example that we've got is, as I understand it, in the state of Alaska, that the seed stock, one has to be collected within 50 miles, is it? Somebody here may know more about this than I do, probably do. And that every year you have to use new material. In other words, you can't overwinter or keep a seed stock from previous things. Again, this is, it would seem to be a fairly precautionary. 50 kilometers? Miles, kilometers, yeah. That's 30 miles, right? So anyway, that's another approach that's been taken by another local state here. And what are your thoughts about that?
I think, yeah, it works for Alaska, but Alaska is very different. They have a lot of coastline, and they have a lot of space to do large-scale cultivation. So I think it's different. Another thing I just wanted to bring up is also the work that has been done on cultivars in Korea, for example, where they're basically growing abalone, and then to feed the abalone, they're growing kelp. And so I had a chance to go out and visit, and They're basically just growing the kelp right next to the abalone. But what the issue they had was that in the summer months, the abalone didn't have enough food as the kelp would, die off and the temperatures got too warm. So they were able to develop cultivars that they selected that were able to withstand warmer temperatures. And that way they were able to extend the food for their cultivated abalone. So there are ways to address these cultivar issues and the warming temperatures using some epigenetics. So basically just selecting for strains or even exposing strains to different lab conditions that will make them more resilient to warmer temperatures.
And I'll go back to thinking more about the standing local genetic variation and how that allows some capacity for adaptation to local conditions. And so one of the things that we're thinking about, similar to that 50-kilometer rule, and we don't have any hard and fast rules yet, but something that we're thinking about is how we manage marine regions for growing kelp to maintain that local adaptive potential. And then thinking, for areas where there may have been dramatic losses of kelp and you no longer have that wild stock available for seed production, then maybe moving towards some type of selective technique. But there are methods that can be used that try and broaden the genetic pool. So there's a lot of different techniques for selective breeding or hybridization that could be considered moving forward, and maybe you have some experience with different.
That's good, yeah. And I know also, in addition, the folks at Woods Hole, who I used to work with, and also UConn, so Scott Lindell and Charlie Arish, they are working on a project to breed basically a strain of kelp that is infertile. So basically selecting a strain that if there is a concern that, your strain would get out into the wild, they are looking for these genetic mutations and then selecting for that. So that's just another way to address the research.
Sure. Another topic that I've dealt with, and mea culpa, introduced species, and I think we mentioned this before, and I personally am dealing right now with a whole bunch of very odd seaweeds from Japan, places like Quilcene, Debob Bay, and so forth, and Sargassum is another one. Really, in my personal opinion, want to really try to avoid introduced species. I had the nori business going here. I had Japanese species here. I would never, ever do that again. It was a huge mistake on my part, mea culpa. But I would say don't go there. And we probably don't need to be really honest. We've got diversity here and things we can play with very much. Another thing I just wanted to mention in passing here was the whole idea, and Betsy, you're involved with this a little bit. We're losing kelp, bull kelp in particular, and there's some slides about losing this kelp in Puget Sound. The efforts to basically biobank or cryopreserve some of these genetic stocks so they don't get lost forever. Squaxin Island is about to disappear. I've been working on just how do you get granophytes and get them in culture and get them in as many different places as you can. Just Noah's Ark, almost kind of an approach to things. So in the future, we'll have that genetic diversity to be able to use for restoration and possible cultivation. Should we have questions from folks? Is that ready to go? Okay, so the microphones are open. I would please ask, I've been reminded, if you have a question, either come bring them up by cards or to the speaker. Don't do like I did and sit in the audience this morning because nobody can hear you, okay? So, have at it.
Hi, my name is Jennifer Clark. I'm from Cascadia Seaweeds in Canada, BC.
Hi, Jennifer. Good to see you here.
Canada.
Another one of these old Nori people here.
I've moved to Kelp now.
Yeah, now you've moved on.
I mean, there's obviously a lot of crossover between BC and Washington State as well. And, you know, one of our biggest things is like where to collect parent material, you know, how far, 50 kilometers is what we are doing currently because there's so much not known about that in BC. I'm wondering what, if you know, what the current like status of the genetic connectivity is in some of the more cultured and like Saccharina, Alaria, Macrocystis, and Nereo. Saccharina and Alaria being a very common seaweed to grow. I'm wondering if you know what the status of like genetic connectivity and diversity, or is that still being determined? I have a second question to you, Simona.
I think maybe the best way to answer that is Betsy and/or Brian, will you work with Felipe? No, the only thing I think we know anything about, I think the only thing we really know much about is Nereocystis, right?
Brian, can you come up to the mic?
Yeah. The answer is we know, actually, not a lot, but we do know some things.
I was making my tea. I wasn't paying entirely attention to the question. What was that?
Genetic connectivity of different species of kelp.
That's commonly cultivated.
Yeah.
Yeah.
Bull
So
kelp, sugar kelp,
Yeah.
these kind of things, here.
What's the question?
Do we know what the status is? Like, is there literature or research that's been done already on understanding how connected some of these populations are? For instance, Alaria marginata in Barkley Sound have been found to have 11 kilometers, and there's different genetic signatures from one population to another. So that has like implications if you're collecting within 50 kilometers of our farms.
Sure. What I know is mostly about Nereocystis. I know that Saccharina has been studied pretty significantly, and there's probably more information about metapopulations and population connectivity of sugar kelp. We know its temporal life cycle in Puget Sound, and it's distributed pretty widely. Sugar kelp is. I think what we know about distribution comes from our resource agency, especially recently with distributions in South and the Central Basin. With regards to bull kelp, most of what I know comes from the Alberto lab in Wisconsin. And the microsatellite analysis done there telling us there's effectively two groups, so a coastal group and an inland sea group, and that there's an area of mixing in Admiralty Inlet. It's been there was a postdoc there that was looking at genetics of, I think, primarily bull kelp, but also some saccharines. And the way she explained it to me was that it is, and Philippe will echo this, that bull kelp has very little genetic diversity within populations. So even a given kelp bed is not going to be very diverse, and it's not going to be terribly different from the one that's some distance away. And that's effectively what I know.
Yeah, and I guess like for some of the like the kelps that have got the pneumatocysts on it, really helps with genetic connectivity as well because they're able to float. So I guess I'm wondering for other seaweeds commonly cultivated like Saccharina and Alaria, like do we have to have, are they more similar within that population because they don't have that opportunity to, you know, drift their genes to another however many kilometers down?
Yeah, well, Saccharina is a much more diverse genus. There's lots and lots of species there. It's relatively young. There's probably, therefore, more genetic diversity. We are talking about cultivar development with bull kelp, for instance, and we look to Philippe for a lot of our questions about what are the numbers that we should be starting with, how do we organize our germplasm, and we're taking our cues really from them, and I think that he feels that all of the things that we are doing are sort of overkill for the diversity that we're dealing with. Yeah, that's just my 2 cents.
I think the answer is we don't know a lot.
Yeah, well, that's why I'm here.
That's why you're here.
I do
So the
have-
real question is, how do we, and this was not, we can hear this is deep stuff, how do we get the answers to that?
Yes.
What kind of techniques can we use? What kind of collections do we need to do? What funding we need to have to answer those questions?
Yeah, I do.
Moving on, I don't know isn't a good enough answer.
Oh, yeah.
And I just wanted to remind people of the really good point that there are really differences among species, like the rafting, that can increase connectivity. And so making sure that we make our efforts to understand diversity broad, because it really can vary among species.
Yeah. Just, not only the fact that there's a world besides bull kelp, please, there's all these other species, and I would offer each one of them have different tricks. In the gametophyte as well as the sporophyte. Yeah. You had another question?
I do. What are some of the current diseases in kelp aquaculture here in the Pacific Northwest that we know of and should be aware of as farmers?
I can tell you about nori aquaculture, but I have no idea about
Kelp.
kelp. Anybody? Kate?
So, no, I was looking more to y'all who are doing it. But there is, there are bacterial diseases that are known from Southeast Asia. And the tricky thing with disease, I think, in seaweeds is that they present pretty similarly. Like the symptoms are very hard to discern, and there's a strong interactive effect with the environment. So you can have a stressor that makes a disease much worse. And so being able to have baseline data, understanding what the disease pressures are, but also the diagnostic tools are really nascent, and also the capacity. Like, we just don't have people that have training to be able to diagnose what the diseases are. And so that's a big issue moving forward, something that we'd like to develop collectively.
Yeah, and just to add, when I was doing the nori business way back when, the diseases that we encountered were all pretty much the same as the ones they have in Japan, but they were all from here. You could go out and find them anywhere. There was, in other words, the diseases didn't come in with what was doing. They were already here. Did I say that? Meg.
First, I want to see if Simona wanted to respond to the question at all, and then I have a question for both of you. I'm just
Yeah, and so also what I have seen is there are folks in Europe working at SAMS, and there's a whole database on seaweed diseases, and I can pull up that database. So there's somebody who you can ship a sample to, and they'll be happy to take it and figure out what disease it is. But yeah, I think, thinking about scaling up and thinking about not having a monoculture of all the same genetics is also important, because you can get diseases, and you don't want your whole crop to just be susceptible to it. So diversifying, I think, is also super critical.
Good point.
I'll stand on my tiptoes a bit, and I think this will have to be the last question because I'm also the timekeeper, so I'm going to cut myself off, but not you.
I got the 4-minute signal.
Oh,
Are you going to talk that long? Go ahead.
So a question for both of you. You both spoke about selectively breeding for heat tolerance. Physically, there has to be a limit for cold water species that would grow here. Can either of you comment on what you think that might be? How high a temperature do you think selectively bred seaweeds could potentially tolerate? And also, if you want to get into the micro of it, the equally important and yet invisible. A long time ago, I was a microbial geneticist, and that was so long ago, I barely remember, but the microbial world and how important that is to the health of the kelp and the ecosystem. There's got to be a heat tolerance threshold there as well. So probably not a lot known yet, but take a stab if you want.
I think, I don't know the numbers off the top of my head, but I think that's a great question. And again, you would have to, back when I was working on my PhD, Charlie had this gradient table where basically we had little petri dishes and we'd grow the little kelp gametophytes in those and look at temperature. So you'd have cold water, colder water on one side, it's recirculating, and then warm water on the other side, and then there's a gradient of like 5 petri dishes, so you have a gradient of temperatures. And then we looked at, male gametophytes, female gametophytes, and then sporophytes. And I mean, that's something that I don't think there's like a clear-cut answer, so I think, we should run some tests and find out, like, what is a temperature tolerant for bull kelp or for alaria or for, you could look at ranges. I know bull kelp does occur in Southern California, so is it the same population? Probably not, but is that one more tolerant to temperature than what's growing up here? All those things could get answered, if there's somebody to take that on. But so that's not a concrete answer, but I think there are ways to address that question. And looking at this, again, it's important to look at the microscopic generation as well as the sporophytes to see if gametophytes have a different temperature tolerance.
Actually, there's been a little, Brooke Weigel has done some of this. There's been some work done. 18 degrees kind of is the magic number that keeps popping up in all this when things go wrong. Anyway, we need to get going, right?
But I think unless
Okay.
I was just going to highlight Brooke's work in that the importance of kind of understanding the differences between life stages and that those tolerance can vary. And Robin, too, is working on that, the life stages. The temperature tolerances vary between gametophyte stage and sporophyte stage.
So if you have any particular questions, yeah, Robin, yeah, right here can probably answer much more questions. But great. I threw some things in here. You got any closing statements or?
I think we just presented more questions than answers, but
That's the whole idea.
it's something to think about.
All right. Thank you all.